الأستاذة الدكتورة لبنى منصور، الأستاذة الدكتورة مريان يسري للدعوة for chairing this session. مؤتمر صحية متميز كل سنة بيكون أكثر تميزا، وبيشرفنا كلنا طبعا نستفيد من المحاضرات very high standard اللي بنشوفها. Next presentation titled Clinical and Molecular Characterization of a Cohort of Children with Congenital Disorders of Glycosylation. Will be presented by Professor Ghada M.H from the National Research Center.
Good afternoon, everybody. You must be exhausted from the very nice and occupied agenda today. I'd like to thank Dr. Marianne for her kind invitation and for excellent organization, and also Professor Lobna Mansour and Professor Ibrahim Shukri. And طبعا it is an annual festival for the pediatric neurology. I'm Ghada Abdel-Salam from National Research Center. I'd like to talk about a very specific type of genetic disorders caused by glycosylation defect or congenital glycosylation defect, because it is a type of glycosylation. You may find hypoglycosylation or hyperglycosylation, but I will talk about congenital glycosylation. What is glycosylation? It is a metabolic disorder. It is abnormal function. Glycans is a sugar, and if we put it with lipid, so it is glycolipid. If we put it with protein, it is glycoprotein. Of course, it is a new type of genetic disorders. It is discovered in 1980s by Yahya Khan from Belgium. They are defects occurring in several cell organelles, cytosol or endoplasmic reticulum, or the endoplasmic reticulum and Golgi intermediate compartment or Golgi apparatus. Of course, they are associated with wide range of clinical manifestations, ranging from polysymptomatic, very mild symptoms, and to very extremely severe phenotype with multi-organ involvement. It was discovered that isoelectric transferrin focusing in the screening for N-glycosylation defect in 1986, after six years of ان احنا نجمع glycogen defect والايزوelectric transferrin focusing for apolipoprotein for all glycan senses disorders. It was the nomenclature, the old nomenclature was CDG 1 and 2, but now the new nomenclature is writing the gene and then not italicized as we used, and also hyphenated by CDG. I mean that we say that CAD CDG. Most of the CDG are autosomal recessive except very minor disorders. They are dominant and X-linked. The main group of glycosylation defect is concerned with N-linked protein glycosylation. They are 60% of the glycosylation defect, followed by the disorders of glycosylphosphatidyl inositol, and then the Golgi homeostasis trafficking and transport. The all-linked lipoprotein glycosylation is only 1% of the disorder. They are very rare. Of course, all the glycosylation defect, they are now 160 disease. And it is emerging. They are discovering every day that, but it is only for now, it is 160. A range of symptoms, they are very wide, from non-immune hydrops fetalis to all the systems, to POSI systems. It involves all the organs of ophthalmological, neurological, hepatic, hematological, with infarction like mitochondrial disorders, cleft palate, kidney involvement. I will start. It is a collective patients from the microcephaly brain malformation clinic in NRC over the 10 years period. Diagnosis was made by whole exome sequencing, confirmed by biochemical testing. We are doing the isoelectric transfeme focusing in NRC. I excluded the cases of dystroglycanopathy and skeletal dysplasia. I will present about 15 or 14 cases. The main clinical features was early onset epilepsy, dysmorphic features, progressive microcephaly, severe cognitive delay, failure to thrive, and the brain MRI for most of the cases showed cerebellar atrophy and delayed myelination. I will talk at first about the Dolly-Cole metabolism. Dolly-Cole metabolism, it is the initial step in the glycosylation where I put monosaccharide or oligosaccharide with Dolly-Cole and as a carrier, and we transport through the endoplasmic reticulum. There are many genes, but I will concern with my cases. It is SRD5A3 CDG. They are ultra rare. It is an ultra rare group of cases. It was first described by Iranian scientist, and it is called Carezi syndrome. It only affects the microphthalmia, cataract, nystagmus, retinal dystrophy, but these cases or this family was described was developmental delay and kyphus scoliosis. Of course, this gene, there are few patients all over the world have been described, and part of them have gastroenterological symptoms like vomiting, failure to thrive, and this is my case. I have a case with failure to thrive, vomiting, diarrhea, arthrogryposis, atrial septal defect, and thrombocytosis. And to my surprise, that the whole exome sequencing reveals that it is SRD5A3. And this case also has skeletal malformation. The second effect is in monosaccharide synthesis. It is what is known as Gap CDG. It is a very rare subtype. Only 6 cases have been reported. CAD is an abbreviation of carbamyl phosphate synthetase and aspartate transcarbamylase and the hydroorotase. They are enzymes are involved in the biosynthesis. A key enzyme in the biosynthesis of pyrimidine and uridine monophosphate, and it is required for the biosynthesis of nucleotide sugars, which are required for the glycosylation process. These cases are treatable. It was published in the literature that many cases have been treated with uridine, but our case, after we have diagnosed it as Gap CDG, she didn't believe that uridine will solve her problem. Of course, she presented with epileptic encephalopathy and regression of milestones, and the brain MRI showed delayed myelination. And she has a poikilocytosis, which is characteristic for the Gap CDG. But although she brought the uridine from Amazon, but she didn't believe that it will cure her symptoms, and she died in the ICU. The second group of disorders is the disorders of glycophosphatide inositol. It is relatively common. There are a number of proteins covalently bound with a glycolipid name, and they are localized in the plasma membrane. These are the GPI biosynthesis carried out in the endoplasmic reticulum, Golgi apparatus, and the cell surface through series of catalytic reactions. I have a case of BGAP1 CDG. The boy is 17 years old. He has only seizures, of course, profound delay, and dysmorphic features, and cerebellar atrophy. Of course, hypogonadism. and these dysmorphic features, and he has a homozygous splicing in the BGAP1. The other one is BGAP3 is relatively common because Dr. Zaki have published about 10 cases with this gene. It is characterized by these facies. It is tinted lip and prominent eyes, high forehead, cleft palate, and they have profound retardation, maybe autistic features. But this is two SIPs. One has profound retardation, the other is a little bit better than her SIP. The brain MRI showed that brain atrophy and mild cerebellar hypoplasia. This part of the glycosylation has seizures and the others has no seizures. This is a very, the family has 4 affected or 5 affected. They have these dysmorphic features and progressive cerebellar atrophy over years, of course, and one of them have cleft palate and partially controlled seizure on 3 antiepileptic drugs. Beta-Q CDG, and the girl has moderate developmental delay. She has only 550, but she could stand with truncal ataxia and clonic seizures, and she has progressive cerebellar atrophy. And the West showed that she has this one of the glycosylation CDG. The other disorder is related to focalization. It is a common form of glycosylation where the monosaccharide is integrated into the terminal fragment of N and O lipid-linked glycans. I have a case of FCCSK CDG. The girl presented with abnormal white matter and abnormal signal in the thalamus and encephalomalacia, and she has uncontrolled seizures. And because of no consanguinity, she was diagnosed that this is an insult, delivery insult. But when we performed the WES, it developed that it appeared that she has a homozygous missense variant in this gene. This type of glycosylation defect has a treatment with either mannose or fucose. But she is a Saudi patient, and she left before giving her the treatment, and she disappeared suddenly. I don't know why. She didn't want to hear that this is a genetic disorder. She was relying that this is an environmental etiology of her case. Disorder of N-linked protein glycosylation. It is a very common. This is the most common type of glycosylation defect. The most famous is PM2 CDG, and is the first described in the literature, and it has now a treatment for this one. We had a case of developmental delay, and she has a characteristic inverted nipples and generalized tonic-clonic and failure to thrive, and the girl died 2 years at the age of 2 years. But even when we brought the hair, the result, the parents didn't come to get the results because they do not want to believe that this is a genetic case. Clinical trials or the treatment for PMM2 CDG is giving galactose or now aldolase reductase inhibitor, and acetazolamide. It was published that it helps in improving the ataxia. And lastly, the recent is FDA-approved gene therapy but it is only a clinical trial. There's no treatment available, genetic treatment available now for the cases. We had also a case of MOX CDG. The girl appeared with a very mild symptoms of mild developmental delay at taxic gait, slight truncal obesity, osteopenia, no seizures, no liver enzyme dysfunction, no organomegaly. And the WES revealed that she has a homozygous missense variant in the Gap CDG gene. All the literature and the cases described in the literature, only 30 cases, nearly 30 cases, they are characterized all by these dysmorphic features, developmental delay, seizures, recurrent infection, visual problem, hepatomegaly, but our patient has a very mild symptoms and signs of the Gap CDG. We had also disorders of Golgi trafficking. We had a case of SOG6 CDG. It is known as Shaheen syndrome because it was described in Saudi families. And they have this dysmorphism, progressive microcephaly, attention deficit hyperactivity, hypertrichosis, and elevated liver enzymes and optic atrophy. They have a very thinning of the corpus callosum, slight white matter changes, and slight cerebellar. I couldn't say that the cerebellum is affected. These are two boys in non-consanguineous family, and they are also didn't want to believe that this is a genetic disorder. It was shown in the literature that they have scoring for the CDG disease disorders. They give scores for severity. And these are all the cases described in the literature with different groups of glycosylation defect. It seems that the PMM2 is reasonable, that the range of severity is between all of them. It's not that severe and it's not that mild, but has a range of symptoms and severity. Conclusions or take-home messages, CDG pose a considerable diagnostic challenges because of the phenotypic variability. It should be ruled out from any unexplained clinical condition of which multi-organ dysfunction is associated with neurological involvement or skeletal findings. Despite the progress made by genetic etiologies, careful clinical assessment of patients still remain an essential step to tell about the natural history of the disease. Limited awareness of CDG is the main reason. That's why it is still underdiagnosed. Thank you very much for your listening. This is the Human Genetics Institute, and these are my collaborators and national collaborators and the international collaborators, and thank you very much.
Thank you very much, dear Professor, and it was very interesting lecture. طبعا انا بشكر الاستاذ دكتور مريان، وطبعا استاذي الكبير دكتور ابراهيم شكري، واستاذتي دكتوره لولو منصور على على تجميع المؤتمر. الحقيقه التوبكس كمان فيري انترستنج، يعني مش بس نيورولوجي، احنا برضو انا مش نيورولوجي بس اتس انترستنج فيها، بحيث فبتخلي عشان كده الناس كتير كله عايز يحضر وعايز يستفيد من المحاضره. طيب may I introduce our next speaker؟ هيبقى فيرتشوال دكتورة مريان فيتشول، مش كده؟ Alexandria, Professor Alexandria Darling. She's going to talk about management of movement disorders in children.